Chapter 4, Tissue Level of Organization. A tissue is a collection of specialized cells and cell products, such as extracellular material and the fluid around the cells. And these perform a relatively limited number of functions. Histology is the study of tissues, and we have 4 types of tissues that will be found in the human body. We are going to look at epithelial tissue, and this will cover the exposed surfaces that, like the outside of your skin, line the internal and external passages and form glands. So if you think of your stomach, it will line the outside of the stomach and it will line the inside of the stomach where it has an internal chamber or passageway. Then we have connective tissue. This will fill internal spaces, provide structural support for other tissues, transport material within the blood, store energy reserves. Connective tissue has a wide variety of jobs, and they aren't always closely related, so one connective tissue may function completely different than another connective tissue. Nervous tissue is the 3rd type of tissue, and this is involved with undergoing contractions to produce movement. Now, movement doesn't have to be a large movement. It could be movement of an organ or movement of food through your digestive tract. We have 3 types of muscles: skeletal muscle, cardiac muscle, and the smooth muscle, which lines your organs. Nervous tissue, the 4th type of tissue, will carry information via an electrical impulse, so this is involved with communication. Tissues combine to form organs. So when you have an organ, if you looked at the stomach, you would find that you have epithelial tissue externally and innermost. Then you're going to have some connective tissue, maybe a few different types in there, some muscle, that would be smooth muscle with the digestive tract, and nervous tissue. We start out as an embryo. Embryonic tissue is formed when the cell, the egg, and the sperm fertilized. They form the zygote. The zygote then develops through mitosis into a number of cells in a hollow ball. And then if you think of it, you take that little ball, or it's a balloon, you push it in on one side, and it forms a C-shaped structure. Now, the C-shaped structure has different layers. The outermost layer of the C will become the ectoderm. Ecto means outside, derm means skin. And this outermost layer is going to give rise to nervous tissue and epithelial tissue that lines the outer layer of the organs. The innermost is the endoderm, inside skin, and this is going to give rise to the epithelial tissue that lines the inside of the organ. And the middle layer is called the mesoderm, and this will give rise to muscle and connective tissue. We're going to start with epithelial tissue. Again, epithelial tissue will also be found internally and externally in organs and will also form our glands. The epithelial layer of cells is going to cover that outermost and innermost surface of the organs, and glands are going to produce secretions, fluid secretions. There are a variety of functions for epithelial depending on where it's located and how many tissue layers it is. Physical protection, if it's an epithelial tissue with a number of tissue layers, and this will provide protection against abrasion, dehydration, destruction by chemical and biological agents. It can also control permeability. Some of our surfaces are going to be relatively impermeable, not allow fluid to move in and out. Others will be fairly permeable. Okay. It will provide sensation, and that's a specialized type of epithelium called neuroepithelium. And this is going to have sensory cells for smell, taste, sight, hearing, and equilibrium. And the last thing is it will produce specialized secretions. That's in the glands, in the glandular epithelium. There are 6 different characteristics that epithelial will have, and these are going to make it specialized to be the outermost or the innermost layer. Okay, the first is cellularity. Your epithelial tissue is composed of closely bound cells. So it's a whole layer of cells, one next to the other. When we look at connective tissue, you'll see that's totally different because the cells are scattered with other material in between them. But with the epithelial, the cells will form like a boundary. And they're going to be closely packed together. They're going to be held together by cell junctions. And they have very little cellular matrix. Matrix is just a fancy word for material. Cellular matrix means there's not much stuff between the cells because they're in tight contact. The second characteristic, contacts. These are going to be your cell junctions, where the cells are going to be linked together so they form a nice strong barrier. Okay, now we want to keep in mind, you want to think of it, especially if it's external in your skin, this is like a fence. You don't want material, you don't want germs and bacteria getting in through the skin. So you want to think of this as a fence. Your cells are going to be close together like the slats of a fence would be. They're going to be held together so there's no wiggle room, so nothing can squeeze between them. Polarity is, if you think of poles or opposite poles like on a battery, it means that two different sides have a different thing going on. With polarity, we're going to have structural and functional differences between the exposed surface and the attached surfaces. The exposed surface is called the apical surface, and in the diagram it's the one that has the cilia and the microvilli on it. This is so it can absorb materials or move materials by the cells. The bottom surface is called the basal surface, and this is going to be attached to a basement membrane, which is will form a barrier between the connective tissue, which lies below, and the epithelial, which is on top. And you can look in those cells and see that there's uneven distribution of the organelles. The nucleus is toward the bottom, the mitochondria are all toward the bottom. So there's different things going on within the cells, opposite poles. Attachment. At the bottom of the cells, you will have the basement membrane. The basement membrane is a two-layer membrane. It's called the basal lamina and the reticular lamina. The basal lamina is produced by the epithelium. The reticular lamina is produced by the underlying connective tissue. Going back to the fence analogy, you got your slats held together well. You don't want anything to get underneath the fence either, so you have to attach it to the ground securely so nothing can crawl under the fence. Okay. The fifth characteristic is avascularity. Vascularity means vessels, blood vessels. You put the letter A in front of it, it means without. So we don't have any blood vessels within the epithelial tissue. Yet the cells still depend on nutrients and oxygen that come from the blood. This is going to be gotten through diffusion from the blood vessels in the connective tissue, releasing their oxygen nutrients up to the cells, as well as from the surface layer, the apical layer, some diffusion will occur. And last is regeneration. Your epithelial cells are constantly replacing themselves. They're going to start at the lower level of cells, if there's a layer of cells, and they're going to move up. And as they move up, they age, and then they get shed and they're dead. Okay. But this, again, go back to your fence. You want to keep it well repaired, so you're constantly repairing any old, worn-out parts. This is not the case with some of our other cells. Again, there are specializations of the epithelial cells. They can move fluids over the epithelial surface. They would do this to provide protection and lubrication, and they'll do this usually with their cilia to move material by it. They can move fluid through the epithelial, and this is to control permeability. Some fluid will move in, some won't, and it varies from which organ we're talking about. And it can produce secretions. And these secretions can provide physical protection or act as chemical messengers. There are two functional regions of the cell. Again, the apical surface is the exposed surface. It will be exposed to the internal or external environment. So when you look at the slide, I'll refer to the free space, because if you look at your skin, the outermost layer, you don't have anything, it's not touching anything, it's touching the air. That's the free space. If you look inside your stomach, the hollow organ part inside where the fluid will be digested, those cells are exposed to that hollow internal organ. Okay. Then we have the basolateral surface. I mentioned basal surface before, but we're going to combine usually the bottom and the sides together and just call them as one surface, basolateral, so bottom and side surfaces. And these will be involved with attaching to other cells on the lateral surfaces or to the basement membrane if it is the bottommost layer of cells in the epithelial tissue. Now we're going to have to maintain our epithelial to keep it healthy, well nourished, and the cells nice and doing their job. We're going to have a number of things, 3 in particular, that we're going to look at when it comes to maintaining the integrity. We're going to look at intercellular connections. Inter, I-N-T-E-R, means between. So between the cells there will be connections. This is going to stabilize the cells so they don't wiggle too much so nothing can squeeze between them. We're going to look at the attachment to the basement membrane. Again, the bottom layer of the cell will attach to the basement layer, so nothing can get underneath the skin, or underneath our fence analogy. And last is just generalized maintenance and repair. If you break a slat in the fence, you got to repair it, or something's going to get in through that spot. If you have a tear in the skin, I'm using that for the epithelial in this case, you will have to repair it so that there is nothing that can get in or out of the tissue. We're going to start with your intercellular connections, and there are a number of different intercellular connections. We're going to break it up into two generalized subgroups, general connections and specific connections. And then we will talk about each of the subcategories under each of those. With your general connections, these are just going to be a widespread connections that are found everywhere, and they don't have a specialized job except for to just stabilize the cells. Okay? And under that we have two different things. We have cell adhesion molecules, also called CAMs. Think of this as a glue. So you take this glue, which is made up of transmembrane proteins, and you just kind of layer it on around the whole cell and then stick the cells together. These are going to be large areas that on the opposing cell membranes, and they're going to bind the cell membranes to each other and to extracellular material and to the basement membrane. The second general connection is going to be a little stronger, and this is an intercellular cement. It is made of glycosaminoglycans, or GACs, and I'll probably use GACs if I ever talk about this. This is sort of like cement. It's stronger. So we've got glue, and then we've got the cement on top of it. This is a thin layer of proteoglycans that's going to be laid down between the adjacent cell membranes. So these two are just very generalized and found throughout all the organs of the body. Then we have specific, very specific connections. These are called cell junctions. And these are going to be specialized areas of the plasma membrane where the cells will attach to each other. They'll have different functions. Under your specific cell connections, we have gap junctions, tight junctions, and desmosomes. The first of the specialized connections is the gap junction. And this is going to be between two cells. They're going to be held together by these interlocking membrane proteins called connections. They are going to be a group of proteins, and if you look at the figure B, I'm showing you the gap junction up close, it's a number of proteins that form a nice little circle, and in the middle of the proteins there is a small channel. And this small channel will allow small molecules and ions to pass through it from one cell to the next. This is going to be used for communication because cells do talk to each other, not the way we do, but they talk to each other using chemicals and ions to let them know what's going on in each cell. They will also allow, in some of our tissues, the connections will work for an electrical impulse to jump from one cell to the next, so we can have a rapid communication of that electrical impulse from the nervous system. Okay. So we are going to see these in a variety of different cells. The second of the specialized connections are tight junctions. Now these are going to be lipid portions of the two cell membranes. They're going to be interlock those lipid portions of the cell membranes with these specialized membrane proteins. Okay, now you want to think of this is if you have two cells, the lipid membrane isn't going to allow fluids to go in and out of the cell. But if they're standing there side by side, there's a little crack between the two cells, so materials can go through there. Maybe water could flow through, or water-soluble molecules. But maybe we don't want anything leaking out of this tissue. So we put these specialized interlocking membrane proteins, almost like a zigzag pattern all the way down, to prevent the passage of any kind of water between those cells. It's going to provide some kind of waterproofing for the cells. And for the tissue. And you're going to find this in organs that don't want to have anything leaking out, even a little bit leaking out between their cells, such as in your urinary bladder. You do not want the urine to leak out between the cells, so you put a new extra layer of protection to prevent that leakage. Also, you don't want it coming out of your intestinal tract. You release the digestive enzymes. You'll just digest all your organs. The third and final type of specialized junctions are called desmosomes. Now these are going to be made up of CAMs and proteoglycans, that intercellular cement. But instead of being a thin layer, we're going to actually put it in large globs on there so we can really hold together certain areas really tightly, and other areas could have a little more flexibility. Now, these are going to be extremely strong connections, and they are going to be found very abundantly in areas exposed to mechanical stress. Think of this as your skin, which is involved with a lot of stretching and twisting when you bend your fingers at each of the joint. That skin is being twisted and stretched, but it isn't ripping apart. So it helps to resist the stretching and twisting of the epithelial that's in the abundant superficial layers of the skin. Now, think about the way to try to think about this anyway. Think about if you had a jacket that had a zipper on, and it was held tightly together with that zipper, so you can't move too much. If you have a shirt on that has buttons, the shirt can twist and torque around the buttons. They still stay attached at the buttons, but that allows for more movement around those buttons. Something you don't get if you have a continual seal. And that is the purpose of these. It allows the skin to twist and torque without tearing, but it still provides a strong connection between the cells. Your desmosomes come in different types. We have button desmosomes, which are little small discs like a button. They are connected to bands of intermediate filaments, and they're going to act as cross braces to stabilize the shape of the cell, so that the cell, even though they twist and torque, they don't change their shape. The second grouping of desmosomes are called hemidesmosomes. Hemi means half, so these are half buttons, and these are going to be found along the basal layer of the cells and the basement membrane. They're going to be attached to the cell with extracellular filaments, and these are going to stabilize the position of the cell and anchor it to the underlying tissues. These are only found in the basement membrane, whereas the other desmosomes, button desmosomes, are going to be found on the sides and the lateral surface. The second thing we're going to do, we've talked about connections. Now we have to talk about how we're going to attach this cell to the basement membrane. Now, your basal surface of the epithelial is attached to this two-part basement membrane. The top layer of the basement membrane is the basal lamina. This is the one that's closest to the epithelial. It is secreted by the epithelial layer, made up of a variety of proteins and other large molecules that they secrete, and it will act as a barrier to restrict movement of proteins and molecules from the underlying connective tissue. The bottom layer of the basement membrane is the reticular lamina. This is the deeper layer, and this is going to be closer to the connective tissue, and this is going to be made up of fibers, reticular fibers in particular, and ground substances secreted by the connective tissue. A ground substance means other things within the fluid and the fibers between the cells. And this is going to help to give the basement membrane its strength. And the last thing we're going to do is just maintenance and repair. Okay, we're just going to keep making new cells. So we use stem cells for that. Your stem cells are going to be cells that are undifferentiated. They're going to be located near the basement membrane. They will produce new cells, and when they divide, they'll produce two daughter cells. One will start to undergo mitosis and grow into numerous cells, and the other one will stay a stem cell so it can replace the old stem cell. And this is allows us to constantly replace the older damaged cells with new healthy cells. We're going to classify epithelial cells in two ways. We're going to classify them by the shape of the cell and then the number of cell layers. So when we name it, we'll have both terms in that name. When you're looking at cell shape, looking at the figure we have above, we have three different generalized shapes. Squamous is thin, flat cells. They look like they've been squashed. Cuboidal looks like a cube, about equal size in height and width, and they tend to have a very large nucleus in the center. And the columnar are like columns in front of a house. Long and thin, they're taller than they are wide, and they tend to have a fairly elongated nuclei near the base of the cell. Also in the figure above is the number of cell layers. We can have either one cell layer, in that case we call it simple, and we can have stratified many cell layers. They have different jobs. Simple tissue will be used for absorption, diffusion, secretion. not for protection. You're going to find these in your digestive and respiratory cavities, your blood vessels, a variety of places. Stratified means there's many layers of cells, and these are going to be for protection. They're going to be in areas that need mechanical or chemical protection, such as your skin, the lining of your mouth, the lining of your anus. All of those are going to have stratified tissue. And you can see we have simple squamous or stratified squamous, simple cuboidal or stratified cuboidal, and simple columnar and stratified columnar in the figure above. Squamous epithelium comes in both simple and stratified. Your simple squamous is going to be a one layer, thin layer of flattened looking cells, and it is going to be involved in absorption, diffusion. It'll form a slick, slippery surface lining certain of the organs. You're going to find it in the alveoli of the lung, lining the heart and the blood vessels, where it's called endothelium, lining the ventral body cavities, where it's referred to as mesothelium. And then you have stratified squamous, and this is going to be found in areas where there's a large amount of mechanical stress. It's going to be found in the skin, the lining of the mouth, the esophagus, the anus. These are just examples of where you're going to find it. Now, we have two different types of stratified squamous, depending on whether or not it is exposed to the external environment where it's drying, or it's exposed to the internal environment where it is moist. If it is exposed to the external environment, it is keratinized. And in that epithelial, the cells will produce a protein that's called keratin that will fill the cells up and minimize water loss to the environment. If it's in the lining of your esophagus, where it's moist and warm in there all the time, it is non-keratinized. This will not dry out, so it doesn't have to produce that extra keratin protein. It's not exposed to the external environments at all, only internally. Cuboidal epithelium also comes in simple and stratified. And again, simple is going to provide very limited protection. It's going to be found in areas where secretion and absorption are occurring. We're going to find these in our kidney tubules. As an example, in the slide you will see in the tissue lab. The kidney is made up of a bunch of tubes, and lining those tubes will be the cuboidal. And if you look at the picture above, you can see in the top picture the cuboidal cells, simple cuboidal, with the hollow space where the tubule is. So it's like cutting a straw in half and you're seeing the hollow space. That's the free space where nothing else is found. We have stratified cuboidal, but this is relatively rare, and we're going to find this in the ducts of our sweat glands and the mammary glands. We do not see that in lab because it is so rare. And then we have a third type that falls under cuboidal, and this is called transitional epithelium. Now, transitional means it changes from one thing to the next. These cells are going to change appearance. They're going to look like squamous at times, or they could look cuboidal at times. And these are found in your urinary bladder. When your urinary bladder is empty, the cells are going to look nice and cube-like. But when it's filled, the tissue will stretch and stretch the cells so they then start to look squamous. You're going to name these in the lab when you're looking at the slides by what the top layer looks like in the tissue that's stratified, because often it will get distorted as you go deeper in. So you want to look at the top layer of the bottom slide, and you can see where it looks cuboidal, or when the bladder is full, they look more like they're flattened cells. Columnar epithelium, again simple and stratified types. Simple will be, again, involved with absorption and secretion, and you're going to find this in your stomach, your small intestines, and your gallbladder. Stratified is relatively rare, just like stratified cuboidal, and it is there for protection. And you're going to find this in parts of the pharynx, which is the throat, the epiglottis, which is the flap that covers the trachea when you are swallowing food, and in some areas of the anus and the urethra. There's a third type under the columnar epithelia called pseudostratified columnar epithelium. Now, pseudo means false, so it's falsely stratified. Now, if you look at the top picture, you can see that there's one layer, and you can kind of figure out that it's all about the same, the nuclei are all in the same layer, and that's how we know there's only one row of nuclei, so there's only one layer, simple columnar cells. But with pseudostratified, the bottom picture, it looks more like there's a bunch of different levels of nuclei, similar to the stratified. Okay. However, the difference here is that they're just one cell, but the nuclei vary from one cell to the next. In some cells, the nuclei will be at the surface, some it'll be towards the bottom. So it's just kind of random. So it looks like there's a bunch of nuclei, a bunch of lines of nuclei, not quite as organized as the stratified. And that's why it's called pseudo-stratified, because it really is only one cell layer, but it looks like more than that when you look at the slide. And you will find this typically in your nasal cavities, trachea, bronchi, and they usually have cilia associated with it. So the cilia can move material past those cells, such as if you breathe in a dusty room and you get dust down your throat, the mucus that's produced in your throat will grab up the dust, and the cilia will help push it back towards the mouth so you can cough it out. When we're looking at the glands, these are collections of epithelial cells that produce secretions. Your cells can be an individual cell, or your glands can be an individual cell up to a complex compound gland. The figure above is just to show that there's a variety of different types of structures. I will not ask you to name what is a compound, tubulo-alveolar gland. I just want you to see that they come in a variety of different shapes and sizes. Now with glands, we have two different types of glands within the body. We have endocrine glands, which are part of the endocrine system itself, and they're going to produce hormones that are released directly into the bloodstream. The figure above you're looking at are your exocrine glands, and these are going to produce secretions that are released to the body surface, directly to the body surface, not into the bloodstream, so they're only going to affect a localized area. Whereas the endocrine, once they get in the blood, they can flow throughout the body. Now when I say to the body surface, that could be externally to like the skin, or it could be released into your oral cavity, your mouth, like your salivary glands do. That's still the surface of the body. And these are all going to be released through ducts, which are tubes. So when you look at the sweat gland, it's a nice coiled little tube deep down into the skin, and it will have a single tube that goes up to hit the surface, and it will release the fluid that way. Okay. And we're going to classify our exocrine glands in two ways: by the mode of secretion, how they do the secretion, how they release it, and the type of secretion that they do release. Mode of secretion is how the secretions are going to actually be released from the cells where they're produced. You have merocrine secretion, and in this case, only the product is going to be released by exocytosis. So you look at the top picture. What we do is we make the product, we put it in little vesicles. Those vesicles go to the surface of the cell, and then they pop and release it through exocytosis. Mucin will be released this way. Mucin, when it mixes with water, will form mucus, and mucin is produced in the digestive and respiratory systems. Also, your sweat glands will release its secretions through merocrine secretion. Apocrine is where, with that secretion, you're going to lose a little bit of the cytoplasm as well as the secretory product. And you'll see this in your mammary glands. If you look at the middle picture, it's like it fills up at the top of the cell, gets filled up with all little particles of the secretion and little drops of it, and then the cell will pinch that top part off and will release the cytoplasm or the cytosol with the secretions in it. It doesn't damage the cell stays intact, it just has lost a little top, and then the cell will grow bigger again. So both your merocrine and apocrine are going to leave the cell intact and functioning. The third mode is holocrine secretion, and this is not going to leave the cell intact. The entire cell is going to become filled with the secretory product, and then the cell itself will burst and release the secretion at the same time destroying the cell. This is found in your sebaceous glands, your oil glands that are located near your hair follicles. The type of secretion is going to vary between serous, which is watery, mucus, which is a mucus secretion, or glands that will release both mucus and the serous. Now, serous glands often release a watery solution that contains enzymes, and you have three salivary gland pairs. And the parotid salivary glands will release their secretions through serous. Mucin, again, once it mixes with water, will form mucus, and this is going to be released from your sublingual salivary glands, as well as glands of the small intestine. And the mixed exocrine gland will have more than one type of gland cell. So they'll have cells that are releasing serous, watery solution, some that will be releasing mucin. So they make two different types of exocrine secretions. And these are in your submandibular salivary glands. The second tissue type is connective tissue, and it's going to connect the epithelium to the rest of the body, so it's going to be below the epithelium. It's going to provide a wide range of jobs. It provides structure, protection, will store energy reserves for insulation. It is involved with transporting material throughout the body and for defense. There are three basic components or parts of the connective tissue. It will be made up of specialized cells, and this is where it really differs from epithelial tissue. Where epithelial, the cells touch each other, the cells do not touch each other in connective tissue for the most part. In between the cells is going to be some extracellular protein fibers, as well as what's called ground substance. The ground substance can vary from fluid to gel to a calcified filler, and the fibers and the ground substance together are going to make up the extracellular matrix, or the matrix. This is going to account for a volume of the connective tissue. Connective tissue is never exposed to the external environment, and it is often highly vascular and contains sensory receptors for pain, pressure, temperature, and other sensations. We're going to classify the connective tissue in different ways. There are three different types of connective tissue. These are just general categories. We have connective tissue proper, where there are many types of cells and extracellular fibers in a syrupy ground substance. And your connective tissue proper is going to be further broken down into loose connective tissue and dense connective tissue. This is going to be based on how much fibers and ground substance there is. In your loose, the material, the extracellular matrix, is going to be loosely put together with some fibers and material. In dense, there is a lot of fibers, so it gets really, really thick and dense between the cells. The second classification is fluid connective tissues. These are going to be cells in a watery matrix, and this will include your blood and your lymph. And the third is supporting connective tissues. These have a less diverse cell population and matrix. They contain much more highly packed fibers, and they're going to be involved in protecting your soft tissues and support the weight of part or all of your body. And this is going to be cartilage and bone. Mesenchyme is embryonic connective tissue. This is derived from the mesoderm, because your connective tissue all comes from the mesoderm. It is the first connective tissue to appear in the developing embryo, and that mesenchyme will give rise to all the other connective tissue types. Connective tissue proper is composed of extracellular fibers, a viscous ground substance made up of cams and gags, and a varied cell population. They are both permanent and non-permanent residents. The permanent residents will be found there all the time. These will include the fibroblasts, which is the only cell that is always present and is the most abundant cell type. The fibroblasts, blasts are builders, so this is going to build fibers. So it's going to form extracellular fibers and secrete the proteoglycans to make the viscous ground substance. They will eventually differentiate into fibrocytes, which are mature cells that are fibroproducers. Adipocytes are your fat cells. Mesenchymal cells are the stem cells for the connective tissue that will produce the other connective tissue cells. And melanocytes produce melanin, which is a pigment which will give color to the tissue. Non-permanent residents will be there sometimes and not other times, and they are all types of leukocytes, which are white blood cells. They're going to come and go as needed. We have a number of different leukocytes. The first one are macrophages. These are very large phagocytic cells. That means they do phagocytosis. They migrate through the tissue looking for any foreign invaders, and then they engulf the foreign invaders and kill them. There are fixed macrophages that can stay for a long time, and free macrophages that come and go and migrate into and out of the connective tissue. The second type of leukocyte is a mast cell. These are small cells near the blood vessels. They're filled with granules of histamine and heparin, and these trigger an allergic reaction and inflammation. There are lymphocytes, which make the plasma cells that produce the antibodies, so your immune system has the ability to produce its own antibodies if given enough time. And then there are macrophages. These include both eosinophils and neutrophils. They're both small phagocytic cells. Your neutrophils are your first responders. They will arrive first at the site of a wound of any kind. And the eosinophils are picky, and they are there for parasitic infections. In addition to the cells of the connective tissue, we have three different types of fibers. They are all protein fibers. Collagen fiber is the most common fiber in connective tissue. It's very thick, like strands of rope. It's strong, and it's very flexible. Reticular fibers are thinner fibers that are similar to collagen, but arranged a little differently, and they form an interwoven network that is tough yet flexible. It tends to stabilize the positions of the blood vessels, the nerves, and also the functional cells of some organs. That makes compartments to house the different cells of the organs in. And the last are the elastic fibers. These contain the protein elastin, which allows the fiber to stretch and then return to its original shape. The category of connective tissue that is called connective tissue proper has both loose and dense tissues. The loose tissues are going to be three types, and they are considered loose because they have few fibers. Only a few scattered around, not as much as the dense, which are packed with fibers. Under your loose connective tissue, you have areolar tissue, which is what we used as an example on the previous slide to show you all the different fibers, all the different cells, both permanent and non-permanent resonance. This is the least specialized connective tissue. It contains all the cells and fibers found in connective tissue, and the fibers are in a loose arrangement. This can distort without any damage and is found in a number of places, including the dermis of the skin, around your joints, your blood vessels, and your nerves. The second type of loose connective tissue is adipose tissue. This is made of adipocytes, which are fat cells. They're going to provide the padding, insulation, shock absorption, and filler around structures. In an adult, we have what's called white fat, and this is not used for heat production. However, in newborns, they have brown fat, and this is burnt specifically for heat production because shivering is not effective in newborns. They don't have that muscle control to shiver, so they can't produce their heat, so they have to burn their brown fat. You do find brown fat in some adults that are found up in like the Arctic area where it's extremely cold, but in general we don't have any as adults. The third type of connective tissue proper is reticular tissue. This is going to form 3D structures. It's going to fill with reticular fibers, which are the thinner fibers, and it is going to support the functional cells of the organ. You're going to find these in lymph nodes, bone marrow, spleen, liver, kidney, where it houses the white blood cells and it compartmentalizes them. Dense connective tissue, three types, are made up mostly of fibers with a few cells scattered about. We have dense regular connective tissue. This is filled with collagen fibers that are arranged in a parallel network so that they are all going in the same direction. These are going to be found in your tendons and your ligaments. Your tendons attach muscle to bone. Your ligaments attach bone to bone. So it's like a rope where all the fibers run in one direction. So when they pull, they exert all that energy and effort in one direction to move the muscle, or have the muscle move the bone that it's attached to, or to move the two bones. Dense, irregular connective tissue is when the collagen fibers are not all going in the same direction. You'll have a patch of them going in one direction, another patch going in a different direction, so they make an interwoven network. There's no consistent pattern, but this provides much more strength and support. to the areas subjected to stress from many directions. This is going to be found in the dermis of the skin. Around the cartilage, we have what's called the perichondrium, and around the bones, we have the periosteum. These are both made of dense, irregular connective tissue. It tends to form thick, fibrous capsules around your organs, such as your liver, your kidney, your spleen, again for protection. And the last of the dense connective tissue is the elastic tissue. This is dense, regular connective tissue, so it's going to have all the fibers parallel, but it's not just made up of collagen, it also has elastic fibers. And the elastic fibers give it the ability to stretch and then rebound. You'll find these in your elastic ligaments that are involved with vertebrate location. In fluid connective tissues, the matrix is a fluid with suspended proteins. There are two different types of fluid connective tissue: blood, which most people don't think of as a tissue, but it's made up of cells with similar functions. Your blood is made up of the plasma, which is the fluid of the matrix, and formed elements such as the erythrocytes, which are your red blood cells, your leukocytes, which are your white blood cells, and your platelets. Those are also called thrombocytes, that are involved with blood clotting. The second type of fluid connective tissue is your lymph. This may not be something you're that familiar with. Paralleling your blood vessels is another set of vessels called your lymphatic vessels, and they pick up all the excess fluid that's squeezed out of the blood vessels due to pressure, go out into the tissue, and then that fluid gets picked back up in the lymphatic system. This lymph is made up of the interstitial fluid that has left those blood vessels, been picked back up by the lymphatic vessels, and ultimately will return it to the blood. But as it goes along in these lymphatic vessels, it will filter the fluid in the lymph nodes and then return it to the blood, therefore looking for any kind of foreign invaders of any sort that shouldn't be there. Supporting connective tissue provides a strong framework that supports the rest of the body. This is going to involve both cartilage and bone. Cartilage has a very firm gel matrix, which is made of chondroitin sulfates, which is a polysaccharide derivative. And the cells are called chondrocytes and are found in little small housing units called lacunas. Your cartilage is avascular, which means it doesn't have blood vessels, so it is very slow to heal. And the cartilage is surrounded by the perichondrium, which peri means around, and chondrium around the cartilage. This has a layer that is an outer fibrous layer of dense irregular tissue and an inner cell layer for growth. Cartilage grows in two different ways, it has two mechanisms. The first is interstitial growth. In this kind of growth, the chondrocytes in the cartilage matrix divide to make more cells, which then secrete more matrix and then enlarges the cartilage from within. Okay. This begins an embryonic phase and continues through adolescence. So think about it this way. If you have a balloon and you add more air into the balloon gets bigger and bigger and bigger. But the outer membrane is the same. That doesn't change. It just gets fatter, but the membrane stays the same. That is what interstitial growth does to your cartilage. Appositional growth is an increase in cartilage by adding new layers of cartilage to the outer surface. So now if you had a balloon, instead of blowing it up, you wrapped it around with a layer and a layer and a layer of some kind of wrapping material. Therefore, it's increasing in size, but it's increasing from the outside. This would be appositional growth. So the cells of the inner perichondrium in appositional growth are going to undergo repeated division, produce chondroblasts. These are immature chondrocytes. Remember, blasts are builders, which then produce the matrix. The chondroblasts eventually mature into chondrocytes, and this will take place mainly during development and may have some minor damage repair in adult cartilage, but not much. As an adult, cartilage damage does not usually get repaired. There are three different types of cartilage. The most common is hyaline cartilage. And this matrix is made of closely packed collagen fibers. It's tough and somewhat flexible. It's usually covered by the perichondrium, except for in your joint cavities, which do not have perichondrium. It is found in joint cavities, in your connections between your ribs and your sternum, at the end of your nose, in your trachea, and again, the most common, the cartilage. The second type is elastic cartilage. This will contain numerous elastic fibers in its matrix. Therefore, it makes it really resilient and flexible. You're going to find this in your outer ear, your epiglottis, your cartilage in the larynx, which is called cuneiform cartilage, which is involved with your voice box. The last is fibrocartilage. This has very little ground substance. Mainly the matrix is densely interwoven collagen fiber. It makes it very tough and very strong and durable. And you're going to find this in the pads between your vertebrae, your pubic bones, or the pelvis. It resists compression, absorbs shocks, and prevents bone-to-bone contact. When you're looking at these three cartilages in the lab, you want to look at the difference in the matrix because the cells are the same. The cells are all in their little lacunas, but the material between the cells varies. When I look at the hyaline cartilage, I think it looks more like jello. The elastic cartilage will have the dark black cartilage fibers, and the fibrocartilage looks like cement between the cells. The second type of supporting connective tissue is bone. With your bone, their amount of ground substance is extremely small. Remember that was the viscous material, fluid between them. It is not there's nothing really viscous. It's mainly calcified material. 2/3 of the matrix is going to be made up of calcium salts, mostly calcium phosphate and calcium carbonate. And then 1/3 of the matrix is the collagen fibers. This is going to allow your bone to be tough, but yet resilient and a little flexible from those collagen fibers. The osteocyte is the mature bone cells, and these are found living in their lacunas, the little housing unit. And their lacunas are going to be organized around blood vessels that branch through the bony matrix to provide the cells with oxygen and nutrients. The canaliculi are cytoplasmic extensions between the osteocytes. They can use these for communication. So imagine if you're living in a brick room, which is surrounded by bricks. You can't talk to anybody else, but you can bore holes between you and the next person over, and you can talk through that little hole. This is the way the canaliculi work. But the cells don't talk with words, they talk with their cytoplasm and variations within the cytoplasm. Covering the bone is the periosteum. This, again, peri means around, osteum means bone. This is made up of an outer fibrous layer and an inner cellular layer, very similar to the perichondrium. Before we go on to another type of tissue, I want to talk about membranes, and that's because membranes are composed of epithelium and connective tissue. There are 4 different membranes. The first is your mucous membrane, which will be coated with secretions of mucus. You're going to find this lining your passageways, your chambers that connect with the exterior environment, such as digestive, respiratory, reproductive, and urinary systems. Also found covering your eyes, areas that will provide protection from the external environment. The surfaces are moist to reduce friction and facilitate absorption and secretion. And the areolar tissue, now remember your membranes are made up of both epithelium and connective, so the connective tissue of your mucous membrane is areolar tissue. And because your mucous membranes are so abundant, we actually give that a special name, and that is called the lamina propria, which is right below the epithelial tissue. The second group of membranes we've already covered, the serous membranes. These are the ones that line the cavities of your ventral cavity. That is going to include the pleural, pericardial, and peritoneal membranes. These are going to be made up of mesothelium and areolar connective tissue. The parietal portion, remember, lines the cavity itself, lines the box. The visceral portion will actually cover the organ. That will give a little space between the 2 layers filled with fluid and will minimize friction between the 2 seroses. It is there mainly for protection. The third membrane is the cutaneous membrane. This is skin, another name for your skin. It will cover the outer surface of the body. This is the only dry membrane that we have. The other three membranes are all wet membranes. And it is made of stratified epithelium with areolar connective tissue and a lower layer of dense irregular connective tissue. And then the fourth membrane are your synovial membranes. Now the previous three all had epithelium and connective tissue. Synovial is just going to be connective tissue. The epithelium they have is an atypical epithelium, nothing that we've talked about before, and it will develop within the connective tissue head and has no basement membrane. The connective tissue is areolar, and this is going to line your joint cavities that are filled with synovial fluid to help protect them. So these membranes are never exposed to the external environment unless there is damage. The connective tissue also forms a framework for the body, and this is made up of what are called fascia. These are connective tissue layers and wrappings that support and surround the organs, and we have three layers of fascia. Looking at the figure, we have starting from the skin and moving inward towards the serosa of the peritoneal cavity, and in that layer we are going to find those three different layers of fascia. Now their function is going to be to provide strength and stability, maintain the relative positions of the internal organs, and to provide a route for the distribution of blood vessels, lymphatic vessels, and the nerves. The three layers are called, the first is the superficial fascia. Now this layer is also referred to as a subcutaneous layer of the hypodermis. Both of those are the same, all three of those are the same thing. And this is going to be found between the skin and the underlying tissues and organs. It's mainly composed of areolar with some adipose connective tissue, and it's going to provide insulation and padding and allow the skin and underlying structures to move independently. The second layer is the deep fascia. This is made up of dense regular connective tissue, and there are a number of layers with all the fibers in one layer running in one direction, and then the next layer running in a different direction. So it's providing a nice, strong support system. This is going to help the tissues resist forces applied from many directions, and it's also going to help form tough capsules around your organs. So your organs are surrounded by this layer also. And then we have the subserous fascia. Sub means below, so it's below the serous membrane, moving back towards the inside of the skin area. And it is made up of areolar tissue, and it lies between the deep fascia and the serous membrane. Now try to think of this sort of like as a layered cake, where you have pieces of cake, but you put material in between to hold those pieces of cake together so they don't fall apart. The serous layers, or the fascia layers, are like the glue that's holding all these layers together, yet allowing a little bit of movement between the layers. The third type of tissue is muscle tissue, and this will provide movement by a contraction of the cells. So the cells will get shorter, and that will cause movement by pulling on something else. Then they can go back to their original length or even longer if need be. There are three types of muscle. The first is skeletal muscle. This is what you think of when you think of muscles. Your biceps brachii, your muscles of your legs, the hamstrings, and the quadriceps. These are all skeletal muscle. And these are made up of large skeletal muscles, cells that are called muscle fibers. They are long and multinucleate cells. They're incapable of dividing, and they contain the mechanical proteins myosin and actin. These two proteins will pull against each other to make the muscle contract, and they also give the striated or striped appearance to the muscle cells. When I say striated, think of it as going across the muscle fiber. It looks sort of like an earthworm. Okay. These are also referred to as striated voluntary muscle because you control the muscles of the skeletal muscle. The second muscle is cardiac muscle. This is smaller branched cells with a single nucleus. They are called cardiocytes or cardiac myocytes. They are also composed of actin and myosin, which produce a striated appearance. But also located in these, where one cell meets the other cell, it looks like they're sewn together, and these are called intercalated discs. These will interlock the cells together, and they're made up of a large number of gap junctions, desmosomes, and intercellular cement. Your cardiac muscle cells have specialized cells called pacemakers that establish a regular rate of contraction. And your cardiac muscles make up your heart. Okay? They are also called striated involuntary muscle because they do have the striping pattern, but you cannot control these. The last type of muscle is smooth muscle. These are little spindle-shaped cells with a single nucleus. They have no striations. They do have actin and myosin, but they are organized completely differently. They are found in your internal organs, respiratory, digestive, walls of the blood vessels, and these are called non-striated involuntary muscles. They don't have the striping, but they are also involuntary. You cannot control them. The fourth type of tissue is neural tissue. This is involved with conduction of an electrical impulse for communication. The electrical impulse is actually called an action potential. This is going to make up your brain, your spinal cord, and your nerves, and there are two basic cell types. Your neuron, which actually transmits the action potential, and this is made up of a soma, the cell body with the organelles in it, dendrites, which are extensions that receive incoming information, and axons, which carry information to the next cell. And the second type of cell is the neuroglial cells, or neuroglia. And these are the support cells for the neuron. They're going to help nourish the neuron, protect the neuron, wrap it with insulation. So they have a number of different jobs. There are a number of different types of neuroglial cells. The last thing to cover on this chapter and on the future chapters is going to be injury to that system. So here we have tissue injury. We need to restore the homeostasis after the tissue has been injured, and this is going to be a two-step process. The first response in any injury is going to be inflammation, and this is a general defense mechanism that's triggered after the injury occurs. Then we're going to do repair and regeneration of the damaged tissue. With inflammation, this is going to be a response to the tissue damage. The injured tissues may now contain pathogens, toxin, waste, chemicals, plus injured cells, so we need to remove that. Okay, and we need to deal with that. So the first thing we're going to do is our mouse cells are going to release a variety of chemicals that will stimulate the inflammation. Mast cells are related to some of the cells of the immune system, and they will stimulate inflammation, which is a natural response to any injury. When the mast cells are activated, it's called mast cell activation. They will start to release histamine, heparin, and prostaglandins. This is going to result in the dilation of blood vessels, so they're going to get larger in diameter, and this will increase the blood flow to that area of the wound. It is also going to increase the permeability of the blood vessels. That means things are going to leak out of it, such as the plasma, and it will also increase your pain sensation. You get an increase in blood flow due to the dilation of the blood vessels and the increase in permeability of those blood vessels. And this is going to result in the tissue becomes red and warm. The blood vessels have dilated now, so they look nice, bright red, and they have warmth in them, so it makes the tissues become red and warm. You become inflamed at the injury site. This is due to the permeability of fluid leaving the blood vessels. But we're also going to have oxygen and nutrients leave the blood vessels to reach that site of injury, and they're going to be used for repair. And then we're going to have waste and toxins picked up by the blood that can get back into the blood vessels due to that permeability. Also, phagocytic white blood cells will be able to get out of the blood vessels and enter into the injury site, and they will start engulfing all the debris and the pathogens and start cleaning it up. After inflammation comes the regeneration. This is the repair that occurs after the damaged tissue has been stabilized and the inflammation has subsided. At this point, the fibroblasts, which are the fibrobuilders, will move into the area and lay down a dense collagenous framework. This is going to be scar tissue. This will be remodeled over time and eventually may assume a normal appearance. Fibrosis is the permanent replacement of normal tissue by a fibrous scar tissue. So if it doesn't resume the normal appearance, it has undergone fibrosis and you have a scar.